we have instituted internships for the MS students. These internships have been structured to give students an opportunity to broaden their horizons. So they're learning principles and concepts of plant breeding in their courses that they're taking in their master's program. And now through this internship, they've had a chance to actually interface with very successful breeders, mainly those here at the University of Illinois in the soybean program, in the sorghum for biofuel program, vegetable breeding, and corn breeding. And in addition, we've given the interns the chance to interface with the seed industry that's very well developed here in the United States. So the interns have spent time with Monsanto Company in various programs that they'll tell you about. They've spent time with Dow AgroSciences, also in various programs that they'll tell you about. So I'm happy to turn the floor over to these individuals that will tell us about their personal experiences in the internship program. So each will be speaking about a certain segment of the internship program to give you a feel for overall the overarching themes and activities. So first you'll be hearing from Gatsen Namutso. He's going to be speaking on the time with Dow AgroSciences. Frederick Awuku will be speaking about time spent with Monsanto during the internship. Collins Gaborvi will be speaking about his experiences in soybean breeding and in corn breeding here at the University of Illinois. And Prince Kapenti will be speaking on his experiences in the University of Illinois soybean breeding program. So with that, I would like to turn the floor over to Collins. I'm sorry, Godson.
Good morning from the University of Illinois, and good morning to everybody. Good afternoon to those in Ghana. My name is Godson Nyawujo, one of the beneficiaries of the internship, and I'm representing on our experiences with Dow AgroSciences. The outline of my presentation will be an introduction of Dow AgroSciences, the activities that we were part of, our experiences at Sydney, Gibson City, and Indianapolis. Dow AgroSciences began as a chemical company in the 18th, and around 1957, they actually started doing things in relation with plants and crops, and in 1997, they started as a full-time agro company in the area of plant breeding, where they come up with new traits and then come up with plants that they sell to commercial farmers. And then on our visit, on the program, we visited their Sydney station. We also went to Gibson City. We went to Indianapolis. But then we didn't go to other places that they have here in the Midwest. We went only to the 3 places that they have here around the Midwest, around the University of Illinois, and also in Indiana. At Dow AgroSciences Sydney, we were led by Dr. Janon Meyer, who introduced us to their breeding program. She told us about how when they come up with traits and the traits get incorporated into plants, it's brought over to the Sydney station. The plant here is maize. It's brought over to the Sydney station, and then they go through the nursery processes, and then they try to raise plants to a point, and then when the season for growing is not compatible here in North America, they send some of their seeds to Southern America, to some countries for continuity until it comes back, and we do a lot of the nursery activities from the point where the trait has been incorporated into the plant until it is finally sent to the production people when it is multiplied as a product. And when we got there, the activities they were undertaking was leaf sampling in relation to the improvement or the the breeding of the maize plant. And what they use these leaf samples for is for marker-assisted selection. So the leaf samples are taken, sent to their lab at Indianapolis, and then the DNA is extracted. Then with the result they get, they send it back to the breeders, and they're able to make their selection based on the genotypic data that is brought from Indianapolis. So we were part of the leaf sampling. We took part in how to sample the leaves, and we were part of how to package the leaves and finally send to Indianapolis. It was a great experience, something we've never done before. We actually had a feel on how to take samples on maize plants and how accurate it should be for marker-assisted selection. Secondly, we were introduced to their pathology lab, where we met Jonathan, who is their pathology lead at Sydney Station. And Jonathan took us through how if they want to breed for a disease-resistant variety, you have to identify the disease in the plant, they isolate it, they put it on an agar, then they multiply it on the agar, then they now blend it, then add it to wheat, and then they allow it to grow on wheat. Then from there, they take it from there and then they inoculate it on the plant for some period for a period, and they allow the disease to show signs, and then they screen for those that are resistant, those that are moderately resistant, and those that are susceptible. And so we were part of the inoculation process. When we got there, they had isolated the diseases, and they raised them on the agar plates and are already incorporated into the wheat bran. So we were part of only the inoculation process, the inoculation processes. So we helped with inoculating the disease not resistant for a good variety that they are trying to come up with, the disease-resistant variety that they are trying to come up with. It was a great experience with the pathology department, and also we had hands on what they actually do, and it helped us in how to set up, how to come up with a disease-resistant variety. And after that, we had a chance with the mechanization group, where they introduced us to how they mechanize, in the sense that they mechanize to reduce costs, to be faster, to also do a large nursery. So we saw machines like the machines that they use in the planting of the field, the machines that they use in harvesting of their nurseries, and the machine you are seeing in the picture is actually the machine that they use to detassel the female plants to prevent them from self-pollinating themselves. So they detassel the female plant and allow the male plants on the maize field or the corn field to send their pollen to the females, so that they are sure that they have a successful process. So the machine up there is the machine that was used for the detasseling process. And so they introduced us to how they also have some machines that they use to spray disease inoculants on plants, and also to select for the plants that are susceptible and those that are also resistant. So it was a great experience with Dow Agrosciences Sydney, and then we enjoyed our time with them. Secondly, the next week we went to Dow Agrosciences Gibson City, where the lead was Dr. Julian Marionellis, and then the station is actually into breeding of soybean. When soybean traits, or when traits are developed for soybean, they are actually they are inserted into the plants mostly at Indianapolis, and then these plants or these seeds are sent to Gibson City, soybean breeding station. And what they do is to do all the nursery processes. They raise the seedlings. They try to rogue out plants that are not what they want to see. And also, when we went there, some of the things that they were doing was tagging. And then tagging here was what they were doing differently that I've never seen before, was to tag every individual plant in any line. So they tag individual plant, and after that, we took samples of all the plants that we tagged, and then these samples were also sent to Indianapolis, where DNAs are going to be taken, and then they are going to be doing marker-assisted selection. And based on the genotypic data, their selection process is clean and clear, and they are actually able to advance with speed, since they are not bulking everything together to select from. They do individual plants, a kind of single seed descent, and they are able to advance individual plants based on what they see in the genotypic data. It was a great experience being a part of the tagging and also the sampling of the leaf tissues for DNA extraction and marker-assisted selection. They also introduced us to their mechanization processes. Just like Sydney, they also have machines. They have machines they used to do almost everything, from planting down to the harvesting of the nurseries, the harvesting of nurseries that they've planted, and also doing other things like spraying and fertilizer application when it is necessary. And that was also an experience with the soybean breeding station, and a great one. Then from there, we went to their station at Indianapolis, where Dr. Lucio M. Joragai was the one who took us, and he took us through their laboratories, where we saw how all the samples that we've witnessed being picked at the various places are brought into their labs, how the DNAs are taken, and how the genotyping is done, and the processes they used, and then from the lab onto the plant get to the greenhouse, how it is advanced, and how at the end of it all they come up with the seeds that they send to the various stations to be tried. And it was also a great experience. We saw what they do, what they told us at Sydney and Gibson City, we saw them do at Indianapolis. And after that, we went for a field day, a demonstration field, where they have all their trials, where they demonstrate what they have to all farmers, or farmers around the world, and to students also. And as part of that, we went to a field where they are trying their Enlist Door product, a product that is that have both resistance to 2,4-D and glyphosate. So they are trying to put these two traits into both corn and soybean, so you could spray both 2,4-D and glyphosate. So, but then the plant will still be on the field without any hurt. So we saw all those experiences. We asked questions, how they came about the traits, what they did to the traits. Is it a synthetic trait? Is it a trait from an organism? And they told us how they came up with them. And so we had time with them, and we asked them questions, and they came out. We also saw their chemical fields, where they are trying some of their chemicals on the plant, since they are both chemical and agro-science company. And it was a good experience. We received the answers that we wanted, and we also asked so many questions as well. Yeah. Then after that, these are our takeaway and experiences that we had with Dow Agro-Sciences. With Dow, we had experiences on corn and soybean sampling, something we've never done before. And then we had our hands on them. We were able to sample, and we were main parts of the sampling process. They told us how to do it, they showed us how to do it, and we did it just like the way they were doing it. It was a great experience. Also, we had a chance to speak to them, or for them to speak to us, about strategies to reduce time factor in breeding programs, using marker-assisted selection, using GBS and single plant advancement procedures. So they're able to reduce the time factor in the genetic gain equation, leading to a great gain process. And also, they showed us the ways to manipulate the genetic equation to get a great genetic gain by the way they genotype to have their genotypic variances, also the way they phenotype to get their phenotypic variances, and all of this, the way they go about it to get a great genetic gain for their traits. They introduce us to it, and to show us how they do all these things. And also, we have learned how to isolate diseases onto agar and then grow them on the agar, how to prepare inoculant from them, and also how to inoculate plants for breeding for disease resistance. And on top of that, we've had a great networking with the world-class breeders in the world currently, and then we have links, people we can talk to, and we now have a kind of a network. Whatsoever we want during our time or on our future breeding programs, we have people that we want to talk to, or at any time at all that we want to speak to. Yeah, that was our experiences at Dow, and we have a great experience and great takeaways that we are taking home. And on that, I want to say a thank you to our mentors on campus, Dr. Rita Mom. She was like a mother throughout the whole process, and then she took us to other places also. Whatever we do, she does with us, and also she was there in any time at all that we have any problem. I want to also say a thank you to Dr. Brian Diaz. He actually held my hands and taught me how to do process on soybean, and then it took me some time, but then he was there to help me throughout the whole process. And also with him, he also introduced us to his breeding program, and also taught us how to continue, or after crosses, how to proceed until you come up with a product. I also want to say a thank you to Dr. Randall Nelson. He also introduced us to his program, and it was excellent, and also to Dr. David Walker. And also to say a thank you to USAID Ghana and to WACI, to University of Ghana, and the team at WACI and University of Ghana, and also a thank you to Soybean Innovation Lab. It was a great experience. It is an eye-opener to the world of breeding, and we'll keep up with whatever we've learned for a future breeding progresses. Thank you.
Thank you so much, Godson. Appreciate that. We like to take questions, but we're thinking to reserve those to the end. So with that, I'd like to introduce Frederick Awuku. And Frederick will be speaking on the interface with Monsanto Company during the internship.
Right, thank
Yes.
you. Good morning from Illinois. Good morning to all viewers, and afternoon to others. I'm Frederick Justice Awuku, and I'll be talking on our experiences with Monsanto. I'll be talking on our experiences with Monsanto. At Monsanto, we visited 3 stations of Monsanto. The first one was in Chesterfield, the second one was in Stonington, and then finally we went to Pharmacity. The whole time with Monsanto, we had Dr. Emilio Oyazabal, who coordinated everything right from Chesterfield to Pharmacity. But at Stonington, we had a lead of the station, which was Dr. Jeff Morpin, and at Pharmacity, we had Dr. David J. Aldex. To start with, I will talk about Monsanto as a company. Monsanto as a company, it has 3 main divisions, which is the technical division, we have the commercialization division, and we have the management who see to the day-to-day running of the activity of the company. At Monsanto, we interacted with various groups that makes up the whole company, which were the human resource. The human resource group basically were people that deals with the recruitment and getting in qualified people into the company. Then we have the discovery group. The discovery group go in and then mine for new genes and new traits that they have to work on. They don't just work on the current trait, but they look ahead, let's say 50 years to come. What will be the need of breeding, or what will be the need of the society? That's what they do. Then we have the breeding group. The main objectives of the breeding group is to carry out the breeding activities, the major breeding activities, the process, needs to make manipulated by the analytic group. Then we have the trait integration. They integrate transgenic traits into the outcome or the product that they get from the breeding group. Then we have the climate, since currently a major concern to crop improvement is climate changes. So Monsanto has a climate group that deals with that aspect of the breeding. Then we have the pathology group that deals with the disease and other related problems. Then we have the international group that coordinates activities worldwide. With Monsanto, one thing that we realize they do is genotyping. What I know of genotyping is that usually we do genotyping on field where we plant a vast number of seeds or produce from our process, maybe F1, F2, or F3, any stage of the breeding pipeline. Then we sample from these leaves. Then after that, we go in there and then select the plant that we need. But with this company, what they do is they genotype the seeds before the seed goes to the field. So they've developed a technique in which seeds can be chipped without destroying the embryo. That means keeping the seed still viable. And they have a system to the left, which is the soybean seed chipper. And that one is used for genotyping soybean seeds. And to the right, we have the corn seed chipper, which is also used in genotyping corn seed before planting. So if you have 2,000 or 10,000 seeds, you are able to genotype all these seeds. If from these 10,000 is only 10 or it's only 100 or it's only 500 that they need to plant, they go ahead and plant just this. And with this technique, they are able to minimize land use. They only plant the seed that they need to plant. Then we looked into their resistant breeding program. In your left, we have a insect-resistant soybean. The first two on your left is susceptible to the insect, and the one on the right is resistant to the insect. We can all see that the one on your right is performing very well, and that will result in a good yield. The one on the left is something no farmer would like to plant on his field. Then we looked at trait stacking. Since crops on the field are faced with different production constraints at the same time, no crop has one production constraint at a particular time. So they have stacked several traits into the maize that we have seen on the right. The one to the left has one trait integrated in there. So from the picture, you can see some patches in there or some spots that shows susceptibility to a particular trait. And the one in the middle has all two traits. So that is resistant to all these traits. The one on the right has not got any of the traits and is showing severe symptoms. Then we looked at mechanization section. With the mechanization, they have various equipment and things that they used in their mechanized farming. First, we looked at the planter. They had a 4-row planter, an 8-row planter, and a vast variety of planters that they use in order to hasten the work on the field, in order to make planting easier, in order to make it faster. Every time they are thinking ahead, to make the work easier and faster for them. Then we were also introduced to the combines. They had a 2-row combine, they had a 4-row combine, and huge combines for their work, because they have large acres of farm land or large acres of research fields that they use for their research activities. Then we have a tractor to your left top, and that tractor, what it does is that they have several attachments that can be attached to the tractor. Like what you see here is for soil analysis. They are able to get the pH of the soil, they are able to get conductivity, they are able to get nutrient, carbon, or anything you can think of is being done at a go. Unlike what we know of, you go to the field and you sample several portions, and you go and then you do, each time you do P, you do N, which is too laborious. With this equipment, they are able to do everything at a go. And then we also have, to your right bottom, we have the detasseler, which they use to detassel corn. So in summary of this slide, I would like to say, why do they need all these tractors and all these machineries? They have large plot of land to work on, large research farms. They have limited time. They want to reduce error. They want to also reduce labor, because if you have thousands and thousands and thousands and thousands of acres of land to work on, you can imagine the cost involved in acquiring the labor to till all this land and to plant your crops right from the beginning through all the processes. We had a visit at Stonington, and with that, we visited the line development program, and then also with supply chain. So with the development program, we looked at pollination of corn. We looked at note-taking on tassel skeletalization. Actually, it was my first time I had to work on note-taking on tassel skeletalization. And we were taught which tassels are skeletal and which are not. So right now, if I go to any maize field, I could be able to tell which are good tassels and which are the bad tassels. And also, we use iPad in field note-taking. Unlike what I'm used to, we go to the field with our books and our pen, and you take your note. When it's raining, what happens? But the iPad and the software they are using to take these notes is a good thing, and I've learned a lot from that. We also looked at the tasseling of the corn, and we also looked at roguing on the purity test field. Roguing is a very important activity on purity test field, because we would want to minimize contamination to the barest minimum. So roguing is done as much as possible before the crop matures. Then we went to the production unit, because one would say, what is the use of doing all this work? We need to get quality seed. You need to get good seed. You need to get disease-free seed. You need to get seed viable to the farmer at the end of the day. So they have a production unit at Farmer City, which is headed by Dr. David. They took us around how they receive the seed, how they treat the seed, they dry the seed, and everything that happens to the seed until it is bagged, and finally the farmer buys the seed from them. In fact, the whole experience on Monsanto is very great for me, in the sense that to get the chance to visit this huge seed company is something that is one in a time. My takeaway on this visit is I realized that there was a great coordination and communication among the various division and breeders of the company, because one division does something, then it passes it on to the next division, then that division also works on the same crop, then it passes on to the next division. So if there is no coordination, there will be disjunction. Their progress will not be connected. Then also we're introducing new techniques in breeding in order to reduce time factor, because everything in the world is bounded by time. We want to try to, as much as we crops have several production constraints, and these constraints keep changing every time, there is a need to fasten the time or reduce the time needed to come out with new varieties so that we can combat these production constraints as fast as possible. And there was also something I learned was about field mapping, because in the field we have distribution of nutrients, and this distribution of nutrients usually are not even, or even the topography of the field is not even. So they were able to map the field such that if it comes to fertilizer application or application of treatment or any other thing to the field, you know the side that needs more application and the side that needs less application. And if in fact there is flooding or any other thing, you know the side of the field to exclude or the side of field to increase or lower the treatment. And we also saw the need for good record keeping for future use. We keep record not for just today, but in 10 years time, in 15 years time, someone might get hold of this data so that the person doesn't have to go to start right from the scratch. Through this internship, we have had connection with world-renowned plant breeders and seed companies. And I think that this part of the internship is something very good, and I would say that I'm much grateful to have this internship and this connection with these various people on this program. We also have an insight into plant breeding as a business, because all I've been thinking about is, after plant breeding, how can I work for this research company? How can I work for this research company? But from this internship, I think that we could begin thinking about plant breeding as a business. Then my general impression about this whole experience, that it has been a great experience for life, being with this internship and the connection with all these important personalities. I'm so much grateful. To end, I would like to acknowledge the USAID Ghana for sponsoring this program, and the USAID, US taxpayers, Dr. Rita Mom. She has done so much for us, and I don't even know how much to thank her. I would like to say thank you to Dr. Brian Diaz, Dr. Randy Nelson, Dr. David Walker, Dr. Eric Dankwa at Waki, Monsanto Company, Dow Agro Sciences, University of Ghana, Waki, and every person who made it a point for this program to come to reality. Thank you.
Frederick, thank you very much for sharing your experiences with us. We're going to move on and hear from Collins. Collins will be telling us about his experiences at the University of Illinois breeding programs.
Good morning once again. I am Collins Gamalibovi, and good afternoon to all others watching me from other parts of the country. And Good afternoon to all our professors and lecturers back in Ghana. I am presenting on soybean breeding program at University of Illinois. Through this presentation, my outline will comprise relation study, experiences I have acquired, and conclusion. Soybean belongs to the family of Fabaceae and with scientific name glycine mass. Some of the importance and uses of soybean include the commercial production of oil. It is also used as a basic component in our food, especially soya milk, and it is used as one of the major components in animal feed, particularly when we are talking about poultry production. The low phosphorus experiment. Why a low phosphorus experiment? Soils with too little phosphorus result to stunted growth, delay in flowering, delay in shoot growth, purple leaf colorations. Secondly, phosphorus is not very soluble and does not move freely in soil water. It is therefore necessary to conduct experiments on genotypes of soybean that will be able to yield well in low phosphorus soils. For this reason, different genotypes of soybean has been put into an experimental setup in a greenhouse to look at how they will respond to both low phosphorus condition and then a normal phosphorus condition. This is a picture showing the experimental setup of how the whole process was carried out. Currently, there are major issues surrounding our soil fertility in relation to phosphorus. Soils may sometimes have high phosphorus rate, but may be fixed, and those with low phosphorus usually result to the deficiencies as I have listed above. Therefore, the need for these experiments, especially for the sub-Saharan Africa, where we have much issue with our phosphorus soils. In addition, if we are to produce crops to yield well and we exceed the concentration of phosphorus that will be needed, then it is going to cause environmental pollution as a result of the phosphorus leaching into our lakes, rivers, and streams. However, the need for this important experiment. Promiscuous nodulation is the second experiment that is also ongoing at the greenhouse in the University of Illinois. Soybean fixes nitrogen through symbiotic association with rhizobium. However, symbiotic interaction between different soybean genotypes and different rhizobia isolates varies widely, both in terms of ability to nodulate and effectiveness in nitrogen fixation. For this reason, a cowpea rhizobia strain has been collected to nodulate soybean to look at the rate at which this nodulation can help with soybean to produce and fix nitrogen in the soil for its usage, and also conserve some of the nutrients that it produces, so that if there is crop rotation, there is going to be enough nitrogen for the next crop to utilize. Back home and in Africa, most of our soils are low in nutrients. And for this matter, this experiment will help to reduce the rate at which farmers buy fertilizer. The experiment is ongoing, and the preliminary results of nodulation indicates that if you look at the picture, almost all the genotypes of about 419 cowpea cultivars that were obtained and put into this experiment, most of them are showing yellowish in leaf or yellow leaf coloration. However, we can see a few of them with green leaf coloration and with this we can assume that there is nodulation ongoing with those with the green leaf coloration. The experiment is still ongoing, and in due time the appropriate results will be published. Some of the experiences we have acquired through this internship include the right experimental setup for our breeding purposes, making it possible to observe meaningful differences to select for crop improvement, making good observation on soybean both on the field and in the greenhouse for effective selection when we are breeding for cultivars. And lastly, we've been able to carry out leaf tissue sampling effectively in soya beans to avoid contaminations. Aside the soybean experiences, I also happen to work with the corn breeding group, especially on pollination or crossing of corn, how to detassel a corn, and how to breed for resistance on corn. In conclusion, I will say that this program has exposed me to practical principles in plant breeding, thereby providing a better understanding as to what goes on the field, the laboratories, and the business aspect of plant breeding in the development of my career as a future plant breeder. On that note, I want to thank you all and acknowledge the following people for their good work done throughout this internship program. I would like to acknowledge USAID Ghana for their immense support and making it possible for us to be here. Dr. Rita Mom, our only mother here. Dr. Brian Diaz, who is one of our major mentors, my mentor. Dr. David Walker, another mentor. Dr. Randy Nelson, and Dr. Eric Dankwa at Waki, Dr. Stephen Moose, who is my mentor when it comes to corn breeding, Dr. Anthony Steady, also a corn breeder, Dr. Martin Bond, also a corn breeder. Monsanto Company for their good work done and their and for their good work done and the warm reception given to us, Dow Agro Sciences and University of Ghana. Thank you all.
Now I'd like to introduce Prince Kapenti, who will be also speaking about his experiences in the University of Illinois soybean program.
Good morning, moderator and audience across the world. I will be presenting on the University of Illinois soybean breeding program. My name is Prince Boete Pente, and I'm one of the MPhil genetics and plant breeding students from the West Africa Center for Crop Improvement, University of Ghana, on this one of a kind in terms of quality internship program at the Soybean Innovation Lab, University of Illinois. It is a singular honor done me to be in front of you to present to you all that I've learned on this internship over this 9 weeks period. This will be the format of my presentation, an introduction, programs of activities, and my learning impressions. Over the 9 weeks, I have worked closely with world-class faculty members as mentors from the Soybean Innovation Lab. Notable among them are Professor Jake Jovic, Professor Brian Diaz, Professor Randall Nelson, Professor David Walker, Professor Rita Mom, just to mention but a few. My presentation, however, today will focus on only 3 of these great professors. Professor Randall Nelson, who I'll present on first, is a curator of the US AID. The first activity I was involved in is the crossing of the glycine soja derived lines. The material I worked on is actually an F6 generation of recombinant inbred lines from crosses that were made between the glycine marks and the glycine soja. The soja parent actually has narrow leaves, while the glycine marks parent has broad leaflets. The soja, glycine soja, is actually noted for its disease tolerance and abiotic stress tolerance, hence the need for it to be used in this cross. And the interesting thing about this cross is that it is difficult when you are crossing the glycine soja with the glycine marks, as opposed to a cross between glycine marks species. Crossing in soybean is completely new to me, and in fact, when I first saw the flower buds, I told myself this is going to be difficult. I couldn't imagine myself being able to do these crosses. But Professor Randy Nelson encouraged me and told me I could do it, and I ventured into it, and I am glad to say that I have been very successful with the crosses I did. The notable things I learned on this program are how to perform these crosses, how to label and tag these crosses, and then how to do a cross-check for successful crosses, and then how to take records. A significant observation I made during the crosses is that whilst we do most of our crosses around 5:30 a.m. to 9 a.m., here in the States, what I experienced was that crosses usually begin at 9:30 a.m. to somewhere around 1 p.m. And of course, I believe it is because of the tropical condition back in Ghana, which will make pollen lose their viability as quickly as possible, hence the need for us to do our crosses much more earlier in the day. How am I going to apply the skills I have learned? There are traditional crops, especially an orphan crop, Vigna subterranea, which is the local name is Bambara granite. It is a crop that is deemed as a crop difficult to cross, I think this experience with this soybean is going to give me the opportunity to develop a research program on that crop, since it is an orphan crop, to develop it. Of course, there are a lot of people, since I work in a research institution, aside schooling, I believe there are a lot of colleagues out there I'll be able to share this experience with, especially train them whilst I'm doing my project in soybean. The second activity I'll talk about is the mapping of QTL for leaf shape. There has been some mapping work on leaf shape in soybean, but glycine soja has a very narrow leaf as compared to glycine max. And the focus of this experiment is to see if there are additional genes or alleles from glycine soja that might not be present in glycine mix. For now, I have been engaged in the sampling, the leaf sampling of leaf tissues for DNA extraction. Again, I've been exposed, had a hands-on practical experience with DNA quantification, something I have never done before. I know back in Ghana, the only available place to do your DNA quantitation is crop research, and that will probably take you about days because it is done one sample after the other. But it was great to experience this fluorometer or this nanodrop, taking all the 96 well plates, and within seconds, I mean, we had our results. It was a very good experience. Hopefully, if all goes well, I will be able to finish up with the GBS work before I return to Ghana. The significant thing I have learned about this program is that, especially with the mapping of QTL for the leaf shape, is that back in Ghana, usually we take our samples and we put them into small pieces of paper on the field before we get to the lab. Sometimes we get to the lab and we realize we have a mix-up of all the samples we've taken, and we have to go back and take these samples. Significantly, what I've learned here is that there's a 96 well plate that you take onto the field where you directly sample into them. And this is really great because it eliminates a lot of error. And for breeding, you don't want to have error in your sample because that will mess up your whole breeding program. Of course, this will give me the opportunity to try and convince my director back home in Ghana so that technologies like these, especially a practical way of taking sampling like this, we can adopt such a practice. My next activity was with Professor David Walker, and this was to give us a holistic breeding experience because fundamentally what we did was to go through the fundamental basics, things that you really need to do to enhance a successful breeding program. And here we went through preparation of stakes, labeling of envelopes, sorting and packaging seeds for sowing. You don't want to put the wrong seeds in the wrong envelope. You want to make sure when here because planting is done on tractors, and those who will be opening the envelopes and be pouring the seeds, if the seeds are not arranged properly, that will mean that that is the first point of error. And once this is messed up, your whole research program could run into challenges. So it was a great opportunity going through these exercises to learn that it is important that the first point is to begin your experiments right, get your stakes right, get your labelings right, get your right seeds into your right packages. And then, of course, we went through other activities, agronomic activities like transplanting, and whoosh, I never could have imagined we will be transplanting soybeans. I had always thought we will usually transplant vegetables. And believe you me, it's been a great experience seeing soybeans being transplanted. Of course, another critical observation that I'll take home it's the hoeing we did. Back home, we have very short hoes, and therefore you need to bend over before you do your hoeing. Here, the hoes have very long poles, so it is very convenient, and you work for so long, and you never feel tired about working. I think what I will take home from Professor David Walker's program, in addition to what I've just mentioned, is that the next generation of plant breeders must be ready to adopt the next generation of new and simple technology or ways of undertaking basic research and experiment. And I see a very good business opportunity in the preparation of stakes and envelopes when I get back home, because WACI trains a lot of plant breeders each year. So that would be a good business opportunity to explore. Even though my previous colleague has spoken about Professor Brian Diaz's program, I wouldn't end this presentation. And I want to mention that I was also involved in Professor Brian Diaz's breeding program on the tolerance to low P phosphorus on soya, and then the promiscuous nodulation experiment. Of course, I'm constrained with time, so I'll just move ahead and then mention the people who have been very grateful. Yes, before I do that, I want to mention that aside breeding, I have also found a new hobby. Biking has been another experience. We've been provided with bikes, and I couldn't have imagined that this is something I was going to love. I had the opportunity to equally repair my own bike when it was broken, and that is a one-time life experience that will remain with me forever. I am very grateful. I would like to acknowledge USAID Ghana, the American taxpayers, Professor Jake Jovic, Professor Herman Bolero, Professor Brian Diaz, Professor Randall Nelson, Professor David Walker, Professor Patrick Brown, Professor Jiman Dankwa, the Monsanto Company, the Agro Sciences, WACI, University of Ghana, and finally, but not least, Professor Rita Mom for making this internship possible. She had to put in a lot of effort, put in a lot of pressure on us. May God bless America and Ghana. Thank you all.
Thank you very much. Okay, I see a question for the second presenter. So that, Frederick, what should be the lower limit in terms of P for soybeans?
Thank you very much for your question. Actually, the P experiment is being carried out in support with a PhD student and a professor. So I am helping with the experimental setup as part of my internship program to learn the practical skills that are required from the whole setup. So I'm sorry to say that much of the questions related to some of these experiments may not be at my domain. Thank you.
Well, thank you very much for staying with us this last hour and meeting this next generation of African plant breeders. I think that probably everyone in the audience is very hopeful about solutions to hunger, malnutrition, poverty for the African continent in the years to come, with scientists like these that are preparing for productive careers ahead of them. So these students have finished a year of coursework. They are near completion of their internship, and they're soon to embark on their thesis research programs. They're looking for sponsors and advisors for these thesis projects, and as well as always making a pitch for future employers, right? So you've met 4 up-and-coming scientists in plant breeding. I'd like to give a special shout out and thanks to some of the people that made this internship possible. There's very many of you that were involved in making this a meaningful experience for these 4 students, particularly Samson Conlon at USAID Ghana, the folks at BHERD at Michigan State University. Like to give a big shout out to Emilio Orizabal, who organized everything for the Monsanto experience, Dr. Tristan Coram, who organized everything for the Dow AgriSciences experience, to the many professors here at the University of Illinois that you heard the interns mention: Brian Deers, Randy Nelson, David Walker, Pat Brown, Jack Juvic, Martin Bohm, Steve Moose, Herman Valero. And also like to give a big thanks to WACI Director Eric Danqua, and also the teaching coordinator Ajaman Danqua, who organized many things on the side from WACI in Ghana. So greatly appreciate also the SIL team that has supported this whole effort. Armand Toussaint is a PhD student in the Department of Economics here at the University of Illinois, and he's been the day-to-day coordinator for the interns. He's done an excellent job with this, and of course we thank Pete Goldsmith, Courtney Tamini, and Dan Baumgarten that have all contributed to making this internship a success for the students. So our sincere thanks and for your support and your encouragement to produce this next generation of outstanding African plant breeders.